it's typical that one will feel the need to bond with another.
Hawak kamay, ikikita, sumasabay sa musika. Gusto kita isayaw ng magagay.
This figure shows sexual reproduction in flowering plant. There are 7 steps. First, the sporophyte bear flower. Second, the flower produce microspore within anthers and megaspore within ovule by meiosis. Third, a megaspore becomes a female gametophyte which produce an egg, and a microspore becomes a male gametophyte which produce sperm. Fertilization results in a zygote. A seed contains an embryo and store fluid within a seed coat. After dispersal, a seed becomes a new sporophyte plant.
Let's learn about its anatomy. First up, we have the sepals, which protects newly formed flower bud. It is most likely leaf-like and collectively known as calyx. Next up, we have petals, which are varied in size, color, and shape. This is to attract good agent pollinators such as bees and wasps. Not to mention that some flowers don't have petals because they are good wind pollinating agents. Next up, we have the stamen, where it's the development site of microspores within the anther or pollen grains. Next up, we have the carpel, where the carpel has 3 parts, which is the style, stigma, and ovary. This part is where the development of megaspores produced by the ovary. Lastly, we have the ovule, which is the production site of megaspores and contain the female gametophytes. Before I go, there is a variation in flower structures, which are divided into 2, where there is a complete flower structure and incomplete flower structure. A complete flower structure would have sepals, petals, stamens, and carpels, while an incomplete flower structure would have none of these. If both stamen and carpels are present in a plant, they are called a perfect or bisexual plant. While if the plant only has 1 stamen or a carpel, it will be an imperfect or unisexual plant.
Before we go any further, let's take a look at the life cycle of plants in detail. For the development of male gametophytes, it is found in the anther of flowers. Anthers usually have 4 pollen sacs, with each of them containing many microspore mother cells. The mother cells then undergo meiosis to produce 4 haploid microspores, and then mitosis before unequal cytokinesis takes place to form 2 cells enclosed by a finely sculpted wall. The immature male gametophytes are now called pollen grains, containing a tube cell and a generative cell. The generative cell undergoes mitosis again to produce 2 sperms, becoming mature male gametophytes, which bring an end to the whole process, called microsporogenesis. And then we come to female gametophytes. A parenchyma cell in the ovary enlarges to become a megaspore mother cell. It then undergoes meiosis to produce 4 haploid megaspores, with only 1 of them being functional, while the other 3 degenerates. The nucleus of the functional megaspore proceeds to divide mitotically until there are 8 nuclei in the female gametophyte. During the formation of cell walls, only cells will be formed, with 1 being binucleate, which are 1 egg cell, 2 synergid cells, 1 central cell with 2 polar nuclei, and 3 antipolar cells. It's the development of new sporophytes. During this phase, the walls separating the pollen sacs in the anther breaks down when the pollen grains are ready to be released. When a stigma is pollinated by the same species, it germinates, forming a pollen tube. And as it grows, the pollen tube passes between the cells of the stigma and the style to reach the microphyle, which is a pore of the ovule. 1 sperm then unites with the egg to form an endozygote, while the other sperm unites with the 2 polar nuclei centrally placed in the embryo sac to form a 3-end endosperm nucleus, which is unique to angiosperms. The angiosperm nucleus then develops into an endosperm, a nutritive tissue that the developing embryotic sporophyte will use as an energy source. The ovule then finally develops into a seed. Last but not least, cross-pollination, also known as heterogamy. Some species of flowering plants, such as grasses and greens, rely on wind pollination, as do the gymnosperms, which is the other type of seed plants. Much of plants' energies goes into making sure the pollen grains actually reach a stigma. The amount of pollen successfully transferred is very high to ensure the reproduction of plants. Most angiosperms rely on animals or insects for pollination.
So we already highlighted important points on this slide. So now, first we'll look at zygote and pro-embryo stage. Zygote and the endosperm become visible. Zygote is small than cytoplasm, and zygote also divide repeatedly to form pro-embryo. Elongated structure called a suspenser, which is transfer and produce nutrient from the endosperm. Next, globular stage. The embryonic cells near the suspenser become a root. At the end, become a shoot. Next point is outermost cell of the plant embryo will become a dermal tissue. The dermal tissue will protect the plant from the desiccation. Besides that, the cell will divide and produce a single outer layer of cell. Next, stomata will open and close to facilitate gas exchange and minimize water loss. Next is the heart stage and torpedo stage. Embryo has heart shape. Cotyledon seeds leaf appear because rapid cell division. Embryo continue enlarge and elongate takes on torpedo shape. Shoot apical meristem responsible for upper ground growth, while root apical meristem responsible for underground growth. Ground meristem, which is function to rise to the bulk of the embryonic interior. Lastly, the mature embryo stage. Plume mold, which is at the tip of the epicotyle and consists of the shoot tip and a pair of small leaves. Hypocotyle contribute to stem development and terminates in the radical or embryonic root. The cotyledons, which is quite noticeable in eudicote embryo and may fold over. Procambium, this time to form the future vascular tissue. And the embryo develops, the integuments of ovule become seed coat.
Now let's take a look at the differences between monocots and eudicots. Monocots only have one cotyledons. Cotyledons absorb other nutrient molecules from endosperm and passes them to embryo. While eudicots, the cotyledon usually store all nutrient molecules the embryo use.
The next part is about fruit types and seed dispersal. There are 6 kinds of fruits, which are simple fruit, compound fruit, aggregate fruit, fleshy fruit, multiple fruits, and dry fruits. The first one is simple fruits. Simple fruits are fruits that develop from a simple ovary or compound ovary that can have one or several chambers. The second one is compound fruit. Compound fruit is developed from a group of ovaries. The third one is aggregate fruit. Aggregate fruit is the ovary formed from a single flower on one receptacle. For example, blackberries. Next, fleshy fruits. The entire pericarp or portion of fleshy fruit is soft and fleshy at maturity. For instance, peaches and tomatoes. The fifth one is multiple fruits. This kind of fruit only has one receptacle and the ovaries fuse to form a large multiple fruit. As a fruit develops, the ovary wall thickens, become the pericarp that consists of 3 layers, which are exocarp, mesocarp, and endocarp. Exocarp forms the outermost skin of a fruit. Mesocarp is often the fleshy tissue between the exocarp and endocarp, and the endocarp serves as a boundary around the seeds. The last type of fruit is dry fruit. Dry fruits are divided by 2, which are green and legume fruits. The fruits of green can be mistaken for seeds because a dry pericarp adheres to the seed within. It is indehiscent because they do not split open. The second one is legume fruits. It is dehiscent because they split open when ripe. Next, we move to the dispersal of fruit. There are 3 kinds of dispersal of fruits, which are dispersal by air, dispersal by ejection, and dispersal by animals. For the first one, dispersal by air. Dry fruits is often dispersed by wind. For the characteristics of the fruits is woolly hairs, plums, and wings. Next, heavier dandelions fruits uses a tiny parachute. Then milkweed pods split open to release seeds that float away on puffy white threads. Next, dispersal by ejection. Usually, the hyacinth fruit is dispersed by ejection. For the characteristics of fruit that dispersed by ejection is, for the first one, the explosion of the seed pods as they burst open and send their seeds flying. Then, the side of the seed pods facing the sun dries faster than the shaded area, causing the partially dry pod to buckle and pop open. The last one is dispersed by animals. The fruits for this kind of dispersal is always ripe, fleshy, and colorful fruits such as peaches and tomatoes. The characteristics of these fruits is hard and cup. This is to protect the seeds and the seeds can pass through the digestive system of an animal and remain unharmed. For the process of this dispersal by animals is, the first one is, as the flesh of the tomato is eaten, the small size of the seeds and the slippery seed coat mean that tomato seeds rarely get crushed by the teeth of animals. The second one is, the seeds swallowed by birds and mammals are defecated some distance from the parent plant. The third one is, squirrels and other animals bury that gather seeds and fruits, bury them some distance and may even forget where they have been stored. The last one is, the hooks and spines of plover, burr, and cocklebur attach a dry fruit to the fur of animals and the clothing of humans. Next, we move to the last part of fruit types and seed dispersal, which is seed germination. There are three conditions to sustain growth, which is sufficient water, warmth, and oxygen. These conditions help to ensure the seeds do not germinate until the most favorable growing season has arrived. Usually, fleshy fruits is germinate. Because fleshy fruits contain inhibitors, they need bacterial action and fire, and the seed coat bursts and the seed germinates. Okay, some seeds do not germinate until they have been dormant for a period of time. What is dormancy? Dormancy is the time during which no growth occurs, even though conditions may be favorable for growth.